Good morning, good afternoon, and good evening, everyone, and welcome to this Inside Scientific, Scientist.com and Sanguine Biosciences webinar titled Leveraging Programmable CRISPR-Associated Transposases for Next-Generation Genome Engineering. I'm Liam Sanio from the events team here at Scientist.com, and I'll be the host today. So today's webinar is sponsored by Sanguine Biosciences, so a big thanks to them for helping to make this event possible. We're very fortunate to be joined by Dr. Sam Sternberg, Principal Investigator and Assistant Professor in the Department of Biochemistry and Molecular Biophysics at Columbia University. In his presentation, Sam will discuss a novel CRISPR system using programmable RNA-guided transposase and highlight its implications for kilobase-scale genome engineering in cell and gene therapies. Alrighty, well, Sam, with that, I'm very happy to hand things over to you. Take it away whenever you're ready.
Great. Okay, thanks, Liam, and thanks everyone for joining us today. Excited to tell you about some of the work that we've been doing in my lab at Columbia University. So I'll hit a little bit of kind of pre-existing CRISPR enzymes and CRISPR tech, but as you can tell from the title, I'm going to really be focusing on a new type of CRISPR technology known as CRISPR-Associated transposases, or Cas for short. So just giving you a little bit of introduction about where I come from. So I did my bachelor's degree at Columbia as well. My PhD with Jennifer Doudna at UC Berkeley, where many of you know much of the early work on CRISPR-Cas9 began in the days pre-gene editing. I had the pleasure of working with Jennifer on writing a popular science book known as The Crack in Creation, where we try to tell the story of the discovery and also some of the emerging application areas. Including some of the kind of controversies around the use of CRISPR in things like human reproduction. And I'm sure many of you have heard the news in that space the last couple of years. I then spent a year working in a biotech company, Caribou Biosciences, where I worked on developing CRISPR reagents for therapeutic applications. And then I've been at Columbia since 2018. As Leah mentioned, I'm in the Department of Biochemistry and Molecular Biophysics. And I'm really excited to share some of the work that the talented students and postdocs in my lab have been doing since that time. So I always like to start with this slide to remind my listeners that CRISPR, of course, is not something invented or engineered in the lab. CRISPR is a biological pathway found in half of all bacterial species on Earth. So in nature, CRISPR-Cas systems provide adaptive immunity to bacteria from pathogenic viruses. And on the left, we're looking at an actual electron microscope image of a bacterium, the big oval, being bombarded by dozens of viral particles decorating the outer surface of the cell. And so these bacterial viruses are one of the most abundant forms of life on Earth. And just like in higher organisms, viral infections cause cell death. And in the context of bacteria, cell death results in the spewing out of dozens or hundreds of new viral particles that can go on to infect other cells in the environment. So when I joined Jennifer's lab in 2009, one of the foundational discoveries in this field had just come out from Rodolphe Berengaud and colleagues, showing that CRISPR-Cas systems provide a new type of immunity to these infections that bacteria use pervasively in nature. Now, how do they do this? Well, I'm going to skip over lots of the details, but as I'm sure most of you know, there are some star players involved that have now since been developed for gene editing technology. One of them is known as Cas9, which is an RNA-guided nuclease that uses the guide RNA shown here in red to target DNA for cleavage. But as Liam's poll alluded to, there are, of course, many other types of CRISPR enzymes that have been studied and described to date. Conventionally, they go by Cas and then a number, where Cas stands for CRISPR-Associated, and the number simply reflects the rough chronology in which these genes and enzymes were described. And so CPF1 was at some point renamed Cas12, and there are many other forms of Cas12 enzymes, Cas12a, B, C, D, etc. And we also know that bacteria use other kinds of nucleases like Cas13 that still use a guide RNA, shown in red, but they target RNA for degradation. And so the hallmark theme of all these CRISPR-Cas systems conventionally is that they protect themselves from infection by using an RNA-guided enzyme to cut nucleic acid from the virus, which can be either DNA or RNA. Here's another way of representing the biodiversity of CRISPR-Cas systems, where every row on this graph represents a unique type of CRISPR found in bacteria. So type 2A, where my cursor is at on the top right, this is the type of CRISPR-Cas system encoding S-pyogenase Cas9, which is used worldwide for gene editing. And what I'd like you to appreciate is that it's one of more than 30 other subtypes of CRISPR-Cas systems. And so when I started my lab in 2018, we were broadly interested to mine this biodiversity to uncover new versions of CRISPR, both because of their biological interest and, of course, their biotechnology utility. And so again, one of the themes of our lab is to spend part of our time over here on the left investigating CRISPR biology, all of the ways that CRISPRs are used in nature as a form of antiviral immunity, but then taking that knowledge to build a toolkit or a Swiss Army knife of tools that we can apply in other cell types to engineer the genome. And as you all know, this kind of workflow going from the biology to the technology has really revolutionized scientists' ability to both study disease and potentially treat disease. And it's worth mentioning that some of the same properties of immune systems, like being very specific or having diverse and distinct mechanisms of action, these are many of the same properties that make for excellent technologies, excellent tools. And so we think that by continuing to mine biodiversity, particularly in the world of immune systems, we can continue to uncover new kinds of technologies. But I'm going to talk about a new link relating to CRISPR, and that is the increasingly interesting intersection of CRISPR systems and transposable elements, often known colloquially as jumping genes. And it turns out that the evolution of CRISPR-Cas in nature is intimately tied to these jumping genes, and it's this interface between CRISPRs and transposons where we both can explain how CRISPR rose on Earth, and we can look for new molecular innovation, new ways that CRISPR systems have been subverted for new activities that are now beginning to allow us to do a new type of gene editing where we insert kilobase-sized payloads into the genome without having to cut the DNA in the process. And that's going to be the focus of the rest of my talk today. So I wanted to start with a little bit of Transposon Biology 101. So these are pervasive elements found in all domains of life. Quite simply, they refer to selfish pieces of DNA demarcated by the line here at the top, which are defined by these boundary sequences shown in triangles that define the boundaries of the mobile DNA, and then one or more genes inside that DNA that encode for enzymes called transposases. And transposases are responsible for cutting and pasting the DNA from one site in the genome to another. And so on the simple schematic on the left, you can see the circles representing the transposase enzyme binding to the left and right ends of the transposon DNA, cleaving it out of its donor molecule to form the synaptic complex, which is then inserted into a new site in the genome called a target here. And I forgot to add the slide, but famously, transposons make up over 40% of the human genome, and in some organisms, over 80% of the genome derives from these selfish and ubiquitous genetic elements. Now, as tools, transposases have been widely used for making transgenic organisms because they're evolved to insert DNA payloads. They don't leave behind exposed double-stranded breaks. They often have minimal requirements for other enzymes in the cell, meaning that they're pretty self-contained, and they've evolved to mobilize and insert very large genetic payloads. We're not talking about small editing of a few base pairs. We're talking about embedding entire genes or multiple genes in the genome through this type of transposition insertion reaction. But one of the downsides that kind of factored front and center in the project I'll talk about today is that, number one, the DNA targeting step shown here on the left for most studied transposons is nonspecific or random. And so what I'm showing here is a representative piece of data for a transposon called Mariner, where we let this thing run loose in bacterial cells and then used high throughput sequencing to map everywhere in the E. coli genome where this genetic payload was spliced. And as I think you can tell from all of the blue lines, there's really no rhyme or reason to the behavior. It simply blankets the entirety of the genome, which means great for making a random insertion and embedding or integrating your payload somewhere, but very poor if it's important for your application where that payload got integrated. And then for similar reasons, because they don't have any targeting control, it's also very difficult to control the number of times that a payload gets inserted in the genome. But we kind of set out with the hypothesis that probably there are transposases that have evolved new types of targeting mechanisms. And that's because these enzymes, these genes, are the most abundant gene that you will find in all of nature. So this graph I'm showing here on the slide is just plotting the abundance of different kinds of genes in hundreds of sequenced genomes. And as you can see in the circle on the top right, transposase genes are completely off the chart here. And so we should think about these as being some of the earliest evolving genes in life, and that they've probably had many opportunities to interface with and co-opt other pathways in the cell. And that's in fact exactly what happened with the biology that I'm going to talk about today, namely CRISPR-associated transposons. So to simplify what these look like, here I'm showing, again, a cartoon of a transposon. But if we start looking at what other genes are hitching along a ride within this mobile DNA, we find CRISPR systems. So this was first noted in 2017, before we knew what these were doing. But if you just used bioinformatics to make predictions or guesses, we saw CRISPR arrays that looked like they would encode guide RNAs. We saw Cas genes like Cas12 that looked like they would encode the enzymes to do RNA-guided targeting. But there was one really big mystery, which is that these CRISPR systems had completely lost the nuclease part. So I told you that conventionally, all CRISPR-Cas systems protect from viruses by cutting nucleic acid, but these CRISPR systems didn't have a nuclease. And so by just looking at these sequences, looking at the genetic architecture, we envisioned that these nuclease defective systems would still be able to use guide RNAs to target DNA. And then we thought perhaps they might work together with the transposase enzymes to integrate DNA. And that by working together, these might insert the DNA payload at genomic sites through a new type of controlled targeting, namely RNA-guided DNA targeting. And so this was like our hypothesis. We'll refer to these moving forward as CAST systems for CRISPR-associated transposons. And we set out to test whether or not this prediction was in fact the case. And so simplifying to a simple metaphor, canonically, we think about CRISPR as being molecular scissors, but these CAST systems we thought might be functioning as molecular glue to paste genetic payloads into the genome. So how do we test this hypothesis? Well, this was work led by a fantastic graduate student in the group, Sana Klompe, who has since moved on to a postdoc at the Pasteur Institute and recently was awarded the Weintraub Graduate Student Award for her thesis. So she identified a candidate CAST system derived from a bacterium called Vibrio cholera. You can see that these are large 30 or 40 kilobase transposons, but we ignored a lot of the things that we thought were not important, and we focused on those three things I mentioned earlier: the CRISPR systems, the transposase proteins, and those conserved ends or boundary elements demarcating the kind of the start and end of the mobile DNA. And so Sana took those different components that we had identified by just gazing at the genome on a computer. We had them synthesized, and then Sana cloned them into vectors shown here in the bottom right, where we had a CRISPR array encoding a guide RNA. We had the CRISPR genes encoding the CRISPR proteins in blue. We had the transposase genes encoding those transposase enzymes in orange. And then lastly, a mini version of the transposon with those triangular sequences that I mentioned earlier, the left and the right ends, and a small payload that we put right in the middle. And the goal was, do these in fact act like molecular glue? Can these enzymes insert the payload from this plasmid into the E. coli genome? And so we designed a guide RNA to target a site in the E. coli genome. And then in this first experiment, we used PCR primers to detect this integration event through PCR amplification. And as you can see in this representative gel on the left, we got a beautifully clean band when all of the components were present. When we scrambled the guide RNA, we saw no band. And if we removed any of the vectors or even made a single mutation in the transposase gene, we lost this band, suggesting that these Cas systems were indeed splicing the transposon into the genome. Now, where exactly were these going? To answer that, we used DNA sequencing, and I'm showing some representative Sanger sequencing data on the slide. And what we found was quite surprising at first. We had somewhat naively thought perhaps the DNA will go into the target site, which is 32 base pairs. But in fact, the DNA payload was inserted 49 base pairs away. So a very characteristic spacing between where the guide RNA matched the DNA and where the actual donor DNA, the payload, was being integrated. And I'll also just mention that there's a hallmark feature of transposon insertions called a target site duplication. And these were present on both sides of the inserted payload, which told us these are bona fide integration products of a transposase. Now, as you all know from the CRISPR field, there's a lot of attention and focus on specificity. And so we similarly needed to understand not just where it's going when we look in a specific part of the genome, but if we take an unbiased approach for deep sequencing all editing reactions or all editing events in the whole genome. And so we adapted a method called transposon insertion sequencing. The details don't matter, but point being that we can detect any integration event regardless of its location. And when we perform this experiment, well, I've already shown you one example of the data for a well-studied random transposon called Mariner. And now if we compare this to a Cas system from V. cholera, you can see how different these behave. Where with the Cas system, our guide RNA was targeting the site marked here with the triangle. And you can see in the blue lines, which represent the sequencing data, that now nearly every single cell that received the payload received it at exactly the site targeted by the guide RNA. And so this told us that these Cas systems are not only RNA guided, but they're highly accurate. They have high target specificity. Now, are they programmable? If we change the guide RNA sequence, can we change where the payload gets integrated? That was what we tested in this experiment, where we cloned a series of different guide RNAs or CRISPR RNAs. So each graph here comes from a distinct experiment where we sampled hundreds of thousands of bacterial cells. And I think you can appreciate that from each graph, when we moved the guide RNA to target a new site in the genome, we saw that blue data giving us the unbiased result. Where did the payload actually end up? Beautifully tracking with the guide RNA that we had cloned. So this really proved to us that like other CRISPR-Cas technologies, Cas9, base editors, etc., these Cas systems are just as programmable because they're reliant on a guide RNA for their targeting. Now, we heard earlier about CPF1 or Cas12a. We've heard about Cas9. So many of you know that there's not just one Cas9, one Cas12a. There are many different homologs, some of which are smaller, more efficient, more accurate. And so we set out to also mine bacterial genomes for more diverse Cas systems. So here I'm showing on the left an evolutionary tree of different families of Cas systems. And then we screened about 24 of these in E. coli. And you can see here that almost every one of these systems is active to some extent, and they have a range of different intrinsic efficiencies where we're plotting efficiency for DNA integration on the y-axis. There's some other interesting biology that I'm not explaining, but you can check out our publications if you want to learn more. When we tested some of the most active systems for specificity, you can see same result as I just showed you. Beautifully clean integration data at the target site. And importantly, if we now challenge these transposases to integrate DNA payloads spanning a large range of sizes, we see that these systems can insert with near 100% efficiency in bacteria with payloads across a 10-fold range in size from 1,000 base pairs all the way up to 10,000 base pairs. And you should keep in mind that for many therapeutic gene insertion efforts, like for cell therapies, we're typically talking about integrating genes that are on the scale of 5 to 10,000 base pairs. And so this tells us that this technology is ideally suited to handling the types of payloads that might be relevant for many of the downstream applications we can envision. Okay, so conclusion number one on how these Cas systems work. How does this new technology differ from a molecular scissors technology like CRISPR-Cas9? Well, the first step is actually quite similar. It's DNA binding by a CRISPR complex. Here, it's known as cascade. This is very similar to the way that you might think about targeting DNA with the dead Cas9 or dCas9. The guide RNA binds to the DNA with base pairing, but instead of cutting, it just sticks around for a very long time until the next thing happens. And in our case, that next thing that happens is the recruitment through protein-protein interactions of the transposases that drag along the donor DNA, shown here in blue. And once all of these different molecular parts assemble, we get the chemical reaction, which is integration without double-strand breaks that have this hallmark feature of occurring about 50 base pairs away from the target site bound by cascade. Now, as biochemists and structural biologists, we've really wanted to understand this both for engineering purposes, but also we want to know how the different components look to inform the ways that we deliver and engineer the system for applications. And so we've teamed up over the years with Israel Fernandez, a talented cryo-electron microscopist at St. Jude's. And here, working with a talented PhD student, Tyler, they determined structures of this first step of the pathway where cascade drags along a transposition protein to DNA. And so in blue are all the CRISPR parts. In red is the guide RNA. And so you can see that this beautiful complex kind of envelops the guide RNA to promote it finding a matching site in DNA. But it has one more important job, which is to drag along the protein shown in green. And the green subunit is critical because that is what ultimately recruits the enzymes that do the DNA integration reaction. And so these all need to work together. And these first structures gave us the clues to begin to understand how all the pieces fall into place. Now, why is this important? Well, I think beautiful to kind of uncover a new type of enzyme that is the first example of an enzyme that can do DNA insertion programmably. So I've already talked earlier about transposases, things like piggyback, TN5, even viral integrases like HIV or what you might use in a lentivirus. These are enzymes that are great at integrating DNA, but they don't have that targeting control. And so we typically would use these to make random insertions like a lentiviral transduction of cell populations like you might use for creating stable cell lines. Great to get the payload in, but not very predictable. And so you'll often have things like genes getting silenced, genes going into essential regions or nearby tumor suppressor genes. And so can we do better than this random behavior? Now, we also know of many enzymes that can integrate DNA at highly specific sequences, things we might refer to as landing pads. And so phage integrases like BXB1, 5C31, transposases like TN7, these are also great at integrating DNA. And if we have the ability to embed the landing pad into the genome where we want, we can control where they put the payload, but you have to do both parts, provide the landing pad and now provide the integrase. And so we see these Cas systems as being the first solution to DNA integration that is inherently programmable, because these evolved to use guide RNAs to specify where they go. And if we've learned anything from CRISPR, it's that guide RNAs can be easily engineered by scientists in the laboratory. Now, why do we want to use these over other CRISPR technologies? Well, for those of you that have kind of been reading deeply in the field on some of the drawbacks of Cas9, well, primarily they result from an undesired requirement on this DNA lesion upstream of the editing products. So double-strand breaks are great at creating variable indel mutations on the left, because this pathway of end joining is highly efficient in most cells, including many human cells. But if we want to have a targeted insertion of something as an experimentalist to choose, we need to put in the blue thing, which means we need a donor template, and we need the cell to instead go down this homology-directed repair pathway. But in general, this HDR pathway is much less efficient. It's restricted to cells that are actively dividing, which means doing insertions in something like a neuron is out of the picture. The efficiency decreases as the size of the payload goes up. We can't control product purity, and so ultimately, in a population of cells, you'll make lots of things you don't want and a small number of things you do want. And then I think most alarmingly, it's been shown in recent years that double-strand breaks can also cause aberrant outcomes like chromosomal translocations and large chromosomal deletions. Now, let me give you one quick example of the consequences of these large deletions. So T-cell engineering is a major opportunity for CRISPR-based technologies, where in the context of immunotherapy, many researchers and companies are using Cas9 to knock out genes like PD-1 or TCRs, while also using Cas9 or lentivirus or other integration approaches to introduce chimeric antigen receptors, with the eventual goal of engineering cell therapies that are uniquely tailored to hunt down cancer in the patient. But this recent study uncovered the fact that if you perform this type of editing in primary human T cells and use more sensitive profiling like single-cell RNA sequencing or long-read DNA sequencing, what you'll find are a non-trivial population of cells, in this case 5%, that have developed aneuploidy, meaning they have lost either entire chromosomes or large swaths of a chromosome because of this aberrant response to DNA double-strand breaks. And so ultimately, the field is really focused on developing next-generation CRISPR tools that can perform gene editing without DNA double-strand breaks or DSBs. And so rather than relying on nucleases, David Liu and others have developed potent kinds of genome editors like base editors and prime editors. These use a nicking form of Cas9 in fusion with other nucleotide editing domains like deaminases or reverse transcriptases to better control the purity of gene editing products by not going through this double-strand break pathway. So these two approaches are extremely well suited to making either single base pair edits or more flexible edits on the small single to tens of base pair scale. And now we think that these CRISPR-associated transposases give us one of the key tools we'll need to make much larger insertions on the kilobase scale. And I think from kind of an engineering perspective, you can see a very interesting breakdown or kind of overlap but separation between a strategy that might engineer new editors using existing enzyme parts that are put together, like a base editor combining Cas9 with a reverse transcriptase. But in some cases, nature has already invented the types of machineries we'd like to use, like these RNA-guided transposases. And so in our lab, we think about kind of putting our minds on both sides of the equation. We can use protein engineering and other methods to kind of invent entirely new things from our pure imagination, but we can also continue to mine nature for interesting new technologies that have already been evolved on Earth before our time. Now, in the last 15 or so minutes of my talk, I'm going to briefly go through two additional stories. One is going to give you a little bit more information on how these Cas systems accurately target the correct sites for DNA integration. And then I'll give you an update on some recent work we published where we've begun harnessing these systems to make programmable DNA insertions in mammalian cells, in human cells. So vignette number one starts with first an expansion of the world of Cas systems. So it turns out that just like CRISPR is diverse and has many different types, Cas systems also have many different types. And so how do they differ? In general, the transposase modules are pretty similar. You see the same TNSB and TNSC genes, but the CRISPR modules use different kinds of CRISPR genes. Cascades in either type 1F or 1B, or Cas12s in type 5K. Now, we've thought a lot about how these different systems compare and contrast. And so comparing the Vibrio cholera cast I've already told you about with this S. hoffmanni cast that Feng Zhang's lab described, we can see that when we target identical sites in the genome, these both show a very nice peak at the target site, as I've shown previously. But the height of the peak for the type 5 Cas system only accounts for about 50% of the total reads. And so where are the remaining reads? Well, we need to zoom in on the y-axis to see that they're actually randomly distributed across the remainder of the E. coli genome, suggesting that these systems still, in the type 5 context, have this untargeted or kind of random transposition pathway. So I'm not going to talk more about why this is the case for type 5, but I want to tell you how we think the type 1F systems have evolved to achieve this very desirable level of fidelity. And so how do we tackle this problem? Well, we did so by focusing not on DNA integration, but actually where in the genome proteins bind. And so to study binding of DNA binding proteins, we used ChIP-seq or chromatin immunoprecipitation. And as you can see here, when we IP'd the CRISPR complex known as cascade and mapped all along the genome where this complex binds, we see a very beautiful peak of enrichment at the target site matching the guide RNA, but hundreds of off-target sites. And if we move the guide to target a new region, we get a new on-target peak of enrichment, but a new spectrum of off-targets. So this was pretty surprising. It told us that cascade is not all that accurate. It's actually relatively promiscuous. It's sampling hundreds of off-targets, but those off-targets are highly reproducible. So if we compare the data between replicate experiments, you see this beautiful correlation suggesting that the off-targets are being repeatedly sampled between experiments. So why is that? Well, these have sequences matching a small portion of the guide, and they have a matching PAM, just like data from Cas9. And so this told us that the initial step in the pathway where cascade binds DNA is surprisingly promiscuous. I've already showed you data suggesting that the integration is very accurate. And so naturally, we hypothesized that something about these downstream steps involving the binding of these other proteins would be the source of added fidelity. And so we focused on two enzymes, an ATPase, shown here in yellow, and the transposase itself, shown here in red, which does all of the DNA integration chemistry. And skipping over many of the controls, I'll just show this graph where we now compared the genome-wide binding between the CRISPR complex at the top and these other two proteins in the middle and the bottom. And what I hope you can appreciate is that at the target site, shown here by the triangle, beautiful overlap of all three molecular machineries, but all of the off targets bound by cascade are now gone for both TNSC and TNSB. Here's a heat map showing the same phenomenon, where the transposase only efficiently enriches at a single site in the genome, which happens to be the on-target site. Here's a Venn diagram giving you a sense for how many potential off-targets are being sampled by the CRISPR complex, of which only 10% bind the next protein, and then just 1 or 2 sites bind the final transposase, allowing us to update our model for how these machineries assemble in cells. And so instead of there being some magical way that from the first moment in the pathway, the on-target is chosen, there's actually what we think of as like a funneling mechanism. In the first step, it's inherently promiscuous, so many sites are sampled, but only some of them successfully recruit the next component, and only some of those successfully recruit the final component and the donor DNA. And so we believe that it's this sequential assembly pathway that explains the source of accuracy or fidelity in the overall editing reaction. I'm going to zip through this, but just to show some pretty pictures, we've continued to look at how these different parts behave in terms of their 3D structure. And so here are cryo-electron microscope images of this TNSC ATPase. They form these beautiful rings with 7-fold symmetry. We were able to identify ATP molecules bound at the interface between those 7 monomers forming the ring. There is a prominent central aperture or hole that goes right through the middle. And not surprisingly, that's exactly where DNA is bound. And so we now have an updated model for how this TNSC protein gets loaded onto DNA that's been identified by the CRISPR complex cascade. And what's been really satisfying with this model is that it really explains that very characteristic spacing that I mentioned earlier, where 50 base pairs always separate the target site that matches the guide and the transposon insertion site. And in fact, if you measure the base pairs through this model, the DNA that comes out the other end of the TNSC ring is exactly 50 base pairs away. Okay, and then for the last 5 or so minutes, I'll give an update on how we've been working through the challenge to repurpose and harness these systems for DNA insertions in human cells. This is work that's been spearheaded by George and Rebecca, 2 great, fantastic graduate students in the group. And I'll just advertise our recent publication, which I think has been added to the handouts tab in the webinar. And in short, the beauty of these Cas systems is that they have this attractive fidelity and this attractive ability to integrate large payloads. The challenge is, unlike a Cas9, which is a single polypeptide, these require multiple different polypeptides. They require a guide RNA, and they require a donor DNA. And so we've had to think really hard about how to optimize the way we express and assemble these systems, the kinds of assays we develop to study them, the choice of which homologs from nature we actually screen and use in other cell types, the way we engineer the donor DNA, and ways that we can maybe streamline or improve the system by designing rational fusions or other strategies to make everything more compact. And just to give you one example of an obstacle we had to work through, we found by testing tagged versions of the different proteins in E. coli that in some cases, like this one bar right here, a single nuclear localization signal on the wrong terminus of one protein could kill the entire integration chemistry from ever happening. So we had to think really hard about where to fuse NLS tags, how to put genes together with 2A peptides without perturbing the need for this molecular complex to assemble in a functional way. I'm going to skip this and just make the point that we've done some experiments to study how the different factors get loaded onto genomic target sites using similar strategies as the ChIP-seq I presented earlier. Here we use something known as CRISPR-A or CRISPR activation. And in brief, it told us that we're getting very nice targeting of endogenous human genes like the Titin gene here that can actually activate gene expression. And so this was a really powerful way that we could begin to suss out parts of our technology that were or were not working. And when we performed ChIP-seq in human cells with a very similar approach as in bacteria, we saw again beautiful evidence of high accuracy binding, where when we studied where TNSC, that ATPase, is going between targeting and non-targeting controls, the only differentially enriched peak between those data sets was the on-target peak at Titin, shown here. But what about integration? And so for integration, we had to go back to our collection of 20 homologous Cas systems to find eCas2, engineered Cas version 2, that was about 40 times better than the Vibrio cholera system we studied. And with further engineering, we reached efficiencies of about 5% on plasmid targets that importantly dropped to complete zero with a single point mutation in the transposase. And so this was a key control demonstrating that these DNA insertions are using the same exact chemical mechanism as what we studied and characterized biochemically and in bacteria. And if we introduced mismatches to the guide RNA, we saw that there was a complete loss of activity, telling us again that these systems look to be quite accurate, although we're now pursuing more unbiased deep sequencing experiments to better study fidelity. And then finally, in the manuscript, we described a really important additional engineering trick to arrive at eCas version 3 that brought our integration efficiencies at genomic target sites up by 2 orders of magnitude into the single-digit efficiencies. And now in this graph, I'm just showing the difference between eCas2 and eCas3 for embedding genetic payloads at 10 different sites across the human genome, where again, we think this is now an important new foundation that we can build upon for further improvements to get from 1% to 10% and beyond. And so I'm not going to show any of the unpublished data to do that, but you can imagine some of the major buckets where we're focusing our attention. One is further improving and streamlining the way we express and deliver all of the machineries into mammalian cells. 2 is using directed evolution and further homolog mining to engineer and identify transposases or Cas systems that are intrinsically more active, they're hyperactive. And then finally, we've been relying heavily on our structures and our biochemical knowledge about how these work to rationally engineer further improvements. And I'll just leave you with one example of the kind of application where we think that these double-strand break independent insertion methods could be really enabling for therapeutic application. So I'm sure many of you are familiar with cystic fibrosis, which results from mutations in the CFTR gene. Now, one of the most common patient mutations is known as delta F508, found in exon 11. And you can find a number of publications in the literature that have used either Cas9, base editing, or prime editing to address and reverse this mutation back to the corrected healthy sequence. The challenge is that if you look at the entire patient cohort that suffer from this genetic disease, there are thousands of distinct mutations that are sporadically found across all of the exons and introns across this very large genic region of the human genome. And so although in theory, you could apply Cas9 or other methods as bespoke solutions to each one of those, there's just not a feasible way to scale that type of therapeutic approach in the clinic because of reasons of cost and just clinical trial design. And so we favor an alternative approach that would allow for a single universal strategy to correct any one of these mutations by actually inserting the full-length cDNA for this gene in the endogenous locus so that it'll be expressed under the native expression level, the native promoter in the right cell types, and it'll be more controlled than a random gene delivery based approach. And it will similarly work for delta F508 as any of these other patient alleles. And so that's the kind of long-term goal that we've been working towards with these Cas systems. And so I'll just end there by thanking the fantastic students and postdocs in my lab that did the work I presented today, a number of our collaborators at Columbia and elsewhere, and our funding sources. And then thanks again to Liam and Inside Scientists for hosting this event, and I'll be happy to take some questions.
Fantastic. Well, thank you, Sam, for a really great presentation. With that, we'll move right on to the Q&A. And as a reminder, if you do have a question that you'd like to ask, you could submit it using the Q&A panel. Sam, to get things started, are these Cas systems at risk of inserting the genetic payload more than once?
Really good question. So, yeah, as many of you may have noticed, I kind of pointed out that instead of interrupting the target site, the insertion happens at a fixed distance downstream. And so there is a risk that if you have prolonged expression of all the components, what's to stop the system from inserting the same payload over and over? It turns out, as something we've published, that it happens extremely infrequently because of something known as target site immunity. The details don't matter, but in short, we don't think that's going to be a common outcome, but we're looking very carefully at it. And then as with Cas9 based editing approaches, I think the classic use will eventually be transient delivery by either mRNA or purified proteins so that you get a very short-term expression, long enough to get your editing product, but not so long that you start hitting off-target sites or very rare tandem insertions.
Awesome. Great answer. I think you mentioned efficiency a little bit towards the end, but is the technology so efficient that the primary editing outcome is homozygous integration?
Yeah, good question. So in truth, we haven't really looked at that in great detail yet, but I think it's almost certain that at 1%, we're going to have very few cells that are homozygous. So right now, the way we quantified our data was just the percent of sequenced alleles that contain the insertion. So those are averaging across the multiple alleles in the cell and all of the alleles in the population. We're now doing things like isolating clones to kind of quantify that on a clonal basis. But, I think rather than looking too deeply at that, we're really focusing our attention on getting the bulk efficiencies into the double digits, as high into the double digits as we can get. And then I think we can start being more methodical about quantifying heterozygous, homozygous knock-ins, et cetera.
Excellent. Nice question here. Do you think there are any opportunities for multiplexing? So, for example, the simultaneous insertion of GFP, RFP, and BFP at specific genomic loci without cross-insertion?
Oh, I love that question. Absolutely. So we had a Nature Biotech paper a couple years ago in bacteria, but just spelling out many of the fundamental capabilities agnostic to the cell type. And we showed that you can do multiplex insertions very efficiently. The beauty of these systems is, again, they're RNA guided, and it's very easy with the particular type of Cas system we're using to generate multiple guides in the same transcription unit. I think with Cas9, sometimes that's challenging, and so you get these clunky tandem U6 promoter-driven sgRNAs. We can kind of nest a bunch of guides all in a single U6-driven transcriptional unit. It spontaneously gets bound by multiple different CRISPR complexes. And then, yes, we've shown that you can do multiplex insertion. And we have furthermore shown that different Cas systems are orthogonal for their payloads. So I've actually written some grants with some cool and fun experiment ideas, like combining two casts. One will put insertion A at site X, and another will put insertion B at site Y, where you'd have complete control over multiple payloads going in kind of user-defined sites. So I think from kind of a synbio and cell engineering perspective, in the future, once we get all these efficiencies higher, there's going to be a lot of fun things we can do to kind of really decorate the genome with diverse payloads. And by the way, there's nothing to stop you from using a Cas to make an insertion here, using a base editor to simultaneously make a base pair modification there. And so we're thinking a lot about the ways to kind of harness all these different editing strategies in parallel.
Yeah, super cool. Really exciting stuff. Bit of a personal question here, but when you started your lab, did you specifically seek to study the Cas macromolecules associated with mobile genetic elements, or did the genome search data lead you to them?
Yeah, really nice question. So we are now doing a lot more of our own bioinformatic mining. The challenge with bioinformatics is you can't really deal with the scale of data and just say, let's look for cool stuff. You usually have to have a specific type of query you're following up on. And I think what's challenging is that the reason Cas systems were invisible for so long is because if you look at the 15 years of CRISPR discovery, it was almost universally looking for a CRISPR array flanked by a CRISPR-associated gene, and always looking for things that are within a few kilobase pairs of each other. These Cas systems have the CRISPR and the CRISPR genes thousands of base pairs away from the transposase genes. And so a neighborhood analysis would never have seen those because they're not as tightly clustered in 1D space. And so now we're trying to think, how can we go outside the box to find things that don't conform to the kind of paradigms or expectations we have today? In the case of Cas, we very much followed others' work that had first uncovered these oddities looking at genomes. And then we did our own bioinformatics and kind of dug deeper and designed the experiments. But now we're really trying to take a much more agnostic, all-encompassing genomics approach, because I hope I convinced you that transposases are so pervasive and they work in so many different ways that there's no doubt we're not going to find transposases that don't fit in any of the buckets I already showed you. And it's just a matter of how do we look for them and study them. And that's something that we're spending a lot of our time thinking about right now.
Awesome. All right, there's a handful of questions here about applications in mammalian cells and eukaryotic cells. How do you envision delivering the Cas systems in vivo, or do you think maybe early translational studies might be, ex vivo editing and writing?
Yeah, I think certainly the same way that all of the kind of therapeutics companies, even with Cas9, which is more easily portable with current methods, they are pretty much all starting with ex vivo therapies because it really simplifies the delivery paradigm. And as you'll hear at any CRISPR conference, delivery is the first, second, and third major obstacle to therapeutic use in patients. So ex vivo mitigates a little bit of that challenge, although it's still not easy. And you still have to be very worried about cytotoxicity of your delivery reagent. But certainly that's where we're going to start. Eventually, we're with the cystic fibrosis work that we're now beginning, we're teaming up with a lipid nanoparticle scientist, thinking about generating new kinds of LNPs that are particularly well suited to accommodate mixed payloads of RNA and DNA. And so we're going to move more towards mRNA delivery, guide RNA, and then, of course, the donor DNA. We're thinking about viral vectors. Right now, the Cas systems are fairly large, so we probably need 2 AAVs. But again, we're doing engineering to see if we can make them more compact. But yeah, we're starting with cells in the laboratory. And I think that's, if we were zooming ahead of the future, that's where we would start with preclinical experiments as well. But I think there's lots of opportunities to piggyback on all the work that's been done with Cas9 and use some of the same approaches down the road.
Cool. Yeah, it sounds like you got a lot of really cool stuff going on. Love to hear it. So you talked a bit about the size of the payload and how it affects the efficiency, I believe. So what was the size of the largest payload that you've ever managed to integrate using Cas?
So I think we went up to 15 in the paper. So there's some data in the recent Nature Biotech paper on that. I had a really cool meeting recently with someone who's using Cas systems in bacteria. This is less relevant for mammalian cells, but I thought it's pretty darn cool. They're basically doing the following experiment. They use conjugation to deliver an entire chromosome from a donor bacterium into E. coli, but they've put transposon ends in the donor chromosome, and they have succeeded to make a targeted insertion of 2.1 million base pairs of DNA using Cas. So that just gives you a sense that, all Cas cares about are the two ends coming together, and the rest of the payload is just up here, it doesn't care. The transposase doesn't care. So I don't think there's probably a physical limit. The limit is defined by getting the DNA into the cell. So in bacteria, you can deliver megabases of DNA through conjugation. That's going to be harder in human cells because I've never heard of someone delivering 1 million base pairs with a virus or plasmid lipofection. But I think the machinery should not have any upper limits on what it can tolerate, provided that the two ends are present and can be synapsed together.
Yeah, that's super cool. Love it. All right. I think in the interest of time, we'll just make this next question the last one. But how do you see CRISPR-Cas9 based editing methods advancing in the future alongside other approaches like prime editing or cast editing?
Yeah, I think if you look at just the phenomenal pace of development in the clinical trial space and in the kind of private industry space, I mean, I think it's pretty staggering that in 10 years, we went from 2013, the first published examples of using Cas9 in human cells in the lab, to now dozens of clinical trials where we're dosing patients in vivo or ex vivo with Cas9. And there's been hundreds of improvements to the technology, higher accuracy, higher efficiency, chemically modified guides that have a longer lifetime in vivo. So I think it's just phenomenal. And it's real. I mean, I don't know if you could have imagined the pace being that quick in 2013. So it's hard to make predictions, but I think, I do think we're going to see fewer uses of Cas9 nuclease in the future. And we're going to see more and more reliance on controlled strategies like base editing and prime editing. And of course, we would hope that cast editing makes its way into the picture as well, because for large gene insertions, we see them as being safer than random viral vector integration. So we're going to keep working hard and try to make that a reality.
Awesome. Yeah. Can't wait to hear more about this. All right. Well, Sam, thanks so much for joining us. It's really been a pleasure having you with us, and great presentation in the Q&A.
Thank you.
Awesome. We'd also like to thank Sanguine Bioscience for sponsoring this event and making it all possible. And last but not least, we'd like to give a big thanks to the audience for participating. And in closing, we hope you enjoyed this Scientist.com and Inside Scientific webinar, and we'll see you again next time. Have a great day, everyone.